Okay. Okay, so before I move into that particular modeling, so first let me tell you that what is the utility of and basic theory. So generally what happens is that the protein folding is a very deeply intricate process. So the 1969 Leventhal first discussed this, and he described it as a paradox known as Levinthal's paradox. This is actually thought experiment. Simply see that we know that the peptide, any protein, it is formed by the peptide bonds, right? It is formed of amino acids, and amino acids are connected to each other through peptide bonds. Now, suppose if I have 100 amino acid, then how many peptide bonds are going to be there?
So 99.
Very good, 99. So that means that obviously because the last amino acid is not forming any peptide bond with any other amino acid, that's a 1 minus. So simply think of this particular thought experiment. So if we suppose have 101 amino acids, then we have 100 peptide bonds. And according to this particular Ramachandran plot, you can see that there are several states that a protein can stay in. Simply think of this, that starting from, so this is 0, this is 1, that means this is 90. So between 90 and 180, there are several phi and psi bond that is possible, right? There's several psi bonds possible. For example, 90.1, 90.2 degree, 91.9 degree. So there are several psi bonds are possible between 80 to 9, between this 90 to 180. So protein can assume any bond that they want. But let's make it a minimum. So let's assume that there are only 3 states for a psi and a pi bond. So we know that there is 100 amine peptide bonds. That means there are 100 psi bonds and 100 phi bonds. So there are total 200 phi and psi bonds. So if you simply think that there are 3 possible angles for each phi and psi bond, suppose 90, 45 and 180, only these 3 are possible. We are neglecting all the others. So even if we consider only 3 possible states, then also there are actually 3 to the power 200 or 10 to the power 95 conformations that are possible. Among this 10 to the power 95 conformations, protein assumes 1, 2, 3, 4, 5, at most 10 or 100 conformations. But a particular protein with a proper structure assumes only a single conformation. Okay. Now, even if we think, now how would a protein know which conformation to take? Because if I have to move through all the conformation, and even if we assume that there is actually 1 quadrillion sample per second, then also the total time taken for a particular protein folding is 60 times more than the age of this universe. But protein folds in milliseconds. Protein take us very fraction of time to simply fold. So how do protein fold so fast? You get the question?
No, sir, I'm a little confused about this confirmations. Sir, can you please explain like
There are,
what is it?
suppose, do you have idea about phi and psi bonds, right? We discussed this phi and
Yes, sir.
psi bond.
Yeah. Yes, sir. Yes, sir.
So
I got
you
it.
understand that between 90 to 180, there are multiple psi bonds possible.
Yes, sir.
Okay, so that part is clear. So let's assume that there are only 3 states, only 3 kind of 3 psi bonds are possible: 90, 100 and 120 degree. Okay, clear?
Yes, sir.
Okay, so if there are 3 possibles, and if we have 200 phi and psi bonds, then how many different conformations can be there? It is 3 to the power 200. Simple math, right?
Okay. Sir, that is the we are counting here the possibility, right? That
Number
how
of
many
different possible conformations. Yes,
Okay.
3 to the power 200. Correct? Clear?
Yes, sir.
So if we simply say that, suppose there is 1 second, 1 second is lost for one conformation. So what would be the time taken for 3 to the power 200 conformation? 3 to the power 200 into 1 second.
Yes, sir.
So if you solve it, it is going to be almost 60 times more than the age of the universe.
Okay, sir. I got it.
Okay, but the paradox is that proteins fold in milliseconds, not even in second, in millisecond. Generally, if it's a very big protein, then they take around some second, 1 second, but if odd point 1 second, but generally they fold in milliseconds. So this is the paradox. Is it clear now?
Yes, sir.
Okay. So the thing is that how do proteins fold so fast? For example, if I simply ask you that, let's take the basic example. Suppose you have 20 crore rupees. Okay. So
Okay, sir.
suppose to count 2000 rupees, you take around 10 seconds. So what would be the time taken to count 20 crore rupees? It would be too high.
Yes, sir.
But the paradox is that time taken to count 20 crore rupees is 1 second. So how do you count so fast? How do you count so fast? Okay. So similarly, how do protein fold so fast? It is because the protein does not go through all the conformation. It is not that protein has to go through all the conformation. It is simply like if I simply take a look at the money, amount of money, I know how much that money is. That kind of thing. So how do they fold? So for that, they explain the folding funnel. So what is folding funnel? Simply think of this funnel. This is the energy funnel. Okay. When the protein is at the top, the energy at the top is highest. Okay. The energy at the bottom is lowest. So this is energy maxima, this is energy minima. So when the protein is present at the top of the funnel, that means it has very high energy. What is going to be the conformation of the protein?
Sir, you said when the protein is in the topmost portion
Topmost
of the funnel,
position. Yeah.
it will be the highest.
The energy would be highest. Yes.
The highest. Okay.
Yes.
So now, can you please repeat your question
Yes.
once?
So what would be the conformation or the topology of the protein? In what state the protein would be in?
Okay, so then it should be in the region, like the red region of that Ramachandran plot. I mean,
No. I mean, would the protein be in a folded conformation or in an unfolded conformation?
Oh, okay, sir. So it should be folded.
Oh, now why? Because it is full of energy. The bond would be broken, right?
Okay, in that sense, yeah. Then the bond will be broken if the energy is high.
Yeah, energy is high, the bond would be broken.
Yes, sir.
That means if you simply heat up the protein would unfold, right? Because the heat would break the bond. So if energy is high, at this most, the energy is at highest position. That means protein is in a complete unfolded state. So to fold properly, the protein has to move down the funnel. It has to move down the funnel to a position where the energy is lowest. And at that particular state, the protein is in a properly folded conformation. Clear?
Yes, sir.
Okay. So now, how it happens? It's very pretty easy, because when the protein simply comes down through this funnel, what happens? The protein is losing its energy, and slowly the bonds are forming, and at the bottom all the bonds are formed. But the movement is not so easy, because it has been seen that the protein does not move smoothly down this particular funnel. Instead, the funnel is like a mountain. What happens in mountain? Suppose if I go to Himalaya, okay, suppose I am at Mount Everest and I want to come down to Kolkata. So does that mean I have to get down always, or do I have to climb several mountains and cross some valleys?
So we have to cross several mountains and valleys.
Right? Because if we simply go to hill, we know even if I'm going to top, there are certain small hills before that, right? So we have to cross those hills, we have to get down, then again climb up a tall hill, and then we have to go up. The funnel is also like that. When the protein folds, first it reaches a local, it's a local minima. What is local minima? Local minima means at that particular location, the energy is at a minima, but that's not the global minima. So when the protein is climbing down the hill at this particular location, what happens? It has a properly, it has some lower energy. So there are some bonds. But again, that bond is not the perfect bond. For example, suppose I have arginine 1, and in general, in optimal condition, arginine 1 should connect with lysine 5 or lysine 4. Okay, it should connect with lysine 4. But at local minima, I see that the arginine is connected to isoleucine 3. Okay, so this arginine 1 and isoleucine 3 connection is not optimal connection. It's a wrong connection. So what happens when such wrong connections happen? The protein again has to climb a hill, and it reaches a location which is known as local maxima. So when it is again climbing a hill, then those bonds which are not optimal bonds, they are going to break, while the bonds that are the optimal bonds, they are going to stay. Again the protein climbs down to reach another local minima, where again some sort of bonding happens. Again it climbs up a small hill where it reaches a global minima, sorry, global maxima. Like this, it passes through several local minima and local maxima to ultimately reach the global minima. So at global minima, the protein connections are perfect. Okay. So how do they do
Yes, sir.
that? The basic thing is that there are hydrophilic amino acid, hydrophobic amino acids. Now, obviously, our cytosol is made up of maximum is water, 70% water. So at first, the beginning, the hydrophobic amino acid, they move away from the water. The non-polar groups move away from the water. They gets buried inside. But that does not mean that all the hydrophobic amino acids are buried inside. There are several hydrophobic amino acids at the surface of the protein, but their number is, or their population is low. If there are 100 hydrophobic amino acids, at the surface there are at most 5, 7, 8, 10 hydrophobic amino acids. Okay, or at most 20. Rest of the hydrophobic amino acids are buried inside. Second, there are hydrophobic effect which is coupled to the van der Waals interaction, electrostatic interaction. All these interactions they have a major role in protein bonding, and they ensure that the protein is having the correct bonding. Okay, so
Yes,
that
sir.
is how a protein actually folds so quickly, because of the effect of all these forces. So this is the thing that we have to mimic. So this is the condition that we have to mimic to make sure that the protein is folding properly. Okay. So this is the basic agenda behind the algorithm that we have developed. Now, so how do we know? Now there are generally three kind of modeling. Okay, I'm going to talk about that later. So first, so when we try to model a protein, we have to understand and we have to know that how do the servers or the softwares assign a particular structure. Now, as I've told you that the major thing that the major thing that actually happens to play a role in this folding is the amino acid, because they all have a proper chemical characteristics. Now, the malic amino acid, that is methionine, alanine, leucine, glutamate, and lysine. These amino acids have high helix forming propensity. They have a condition so that they trigger to form alpha helical. They have alpha helical nature, okay, and they form alpha helix. But two amino acids, proline and glycine, they break the helix. They are known as helix breaker. But why do they break the helix? First, because proline. How is proline different from all the other amino acids?
Sir, they are non-functional.
They are non-functional. No, no, no.
No, like it's not coded
Huh?
by the it's not coded by the functional genes.
Mm-hmm. All are coded because these are natural amino acids. What's the basic structural difference between proline and the other amino acids? Very easy. Simply think of the structure.
Hmm. So generally in proline there is a I think it has a pentane structure. Matlab, it has a benzene kind of structure, as I remember. I exactly don't remember the structure right now, but it has some benzene kind of structure at the end.
Okay, just go through the amino acid structures. Okay, because protein is all about amino acids. Okay, the major difference between that the thing that proline lacks is that it does not have a free amide hydrogen bond. It doesn't have a free amide bond. It doesn't have a free amide bond, and due to this lack of amide bond, it does not participate in any hydrogen bond formation because it doesn't have a free amino group. And without a free amino group, you can't participate in hydrogen bond. That's the first. It does not have the power or the tool to participate in hydrogen bond. That's one thing. And what about glycine? The glycine is so small. It is so small that it is highly flexible, and this flexibility disrupts the helicity because the molecule is so flexible that it is not able to fix at any standard chart. Because what happens, the glycine does not have a side chain, it has hydrogen, right? It does not have a side chain, so that is why it is highly flexible. So if you simply keep it at a helix, due to the flexibility, it breaks the hydrogen bond. And proline, it doesn't participate in hydrogen bond. So this is the two reasons that they break up the helix. Okay, so whenever you have proline and glycine, you make sure that they generally break up the helix. It's not mandatory that they will break the helix, but generally they are helix breaker. Okay, whereas other amino acid like tyrosine, phenylalanine, tryptophan, all of these have large aromatic residues, the benzene groups that, as you were saying, that is present in phenylalanine, tyrosine, tryptophan. Okay, and this particular threonine, valine, isoleucine, they all have beta sheet forming propensity. So if there is a particular domain which is rich in tyrosine, phenylalanine, tryptophan, then it is going to participate in beta sheet formation. Whereas if you have a domain which is rich in malic, a lot of methionine, lot of leucine, lot of lysine, then you know that particular thing, that particular domain is going to fold into a alpha helix. Clear?
Yes, sir.
Very good. So now using these two things, we generally try to predict the protein structure. So how the protein structures are predicted? First, the backbone of a protein is predicted. How do you predict the backbone? Now there are three different modes of prediction. First is the ab initio method of prediction. Second is the homology method of prediction, and third is the protein threading method. Now, what is ab initio method? Ab initio method means from the scratch. And what do you mean by from the scratch? From
So
the
means from amino acid.
Yes, that means starting from the amino acid structure. They only take into account the amino acid sequence, nothing else. Only the amino acid sequence. They simply take the information from amino acid sequence and they fold the protein. That is the information that they want. That is ab initio prediction. So what is homology modeling? Homology modeling means when you are taking the help of some other proteins which look like you. For example, generally if you simply take a look into me, people say that I look like my father. So when I walk, how the way I talk, the way I walk, or the way I look, I represent my father. So if anyone has to know that how my father looks like, they're going to take my face and then they're going to add extra agedness, oldness, white here, and bingo, my father is there. So this is the homology modeling, when you are not taking into account only the sequence, but you are taking into account the structure of other homologs, the other relatives. Because what happens, most of the protein, they have relatives. For example, Acha, first tell me, what do you mean by homology? What is homology? What does this word mean? Homolog or homology?
Sir, when the structural origin is the same, and
Very
it's
good.
called homology.
Very good. Can you give me name of two proteins which share similarity?
So right now I don't remember
Okay.
exactly.
Acha. Then, okay. Can you tell me a protein in what's that called? In glycolysis, which participates in glycolysis? Hexokinase. Remember hexokinase?
Yes, sir.
So what does hexokinase do?
No idea. I'm currently just black out of the whole situation. I don't know.
Okay, it's a very easy. It participates in glycolysis. Okay, so hexokinase is a homolog, that is glucokinase. It is present in liver. So what is there? That there are 90% similarity between hexokinase and glucokinase, and there are 10% dissimilarity. So if I have to structure glucokinase, what I'm going to do, I'm going to take a look into the hexokinase, and I'm going to know that is 90% of the protein structure is similar. So I have to predict the rest of the 10%. So that is the homology modeling, where you take the help of other proteins. Similarly, there are multiple proteins that are present in our body that has other homologs, as that are present in plant cells, that are present in bacterial cells, that are present in viral cells. For example, the protein, suppose this hexokinase protein that is present in me, and the same hexokinase protein is present in cat, dog, plant, tree, you name an animal, it is present there. So if it is present on those, does the structure is same? No, the structure is not exactly the same. Me and my father, we are not exactly the same, but there are similarity. So that is homology. There are similarity between some proteins. So we have to trap down those similarity, and we have to simply check which protein has that similarity, and then find the similar molecules, and then we have to see whether that particular structure is solved. And if it is solved, then we take that particular domain whose structure is solved, and we try to manipulate the other unknown domain. So this is homology modeling. Clear?
Yes, sir.
And next comes the protein threading. In protein threading, it's absolutely a different scenario, where what happens, they are in protein threading, you simply break down the entire protein into few fragments. Suppose it's a 100 amino acid protein, then what do, you simply break down the protein into several, suppose 10 fragments, then each of the fragment is consisting of 10 amino acid. So now you model 10 amino acid stretch individually. You model all the proteins individually, you try to model them, and once you model them individually, then you simply stitch all the domains together to get a complete shape. For example, if you have you seen how a Durga idol is made?
So not really, but I know
But
a
what
little.
happens generally is that they simply get a total entire thing, and later you know that uh, in pottery, or in when we make a clay model, what happens is that we simply try to make everything separately. First we shape up the face, okay, then we make a body, we attach the face to the body, then we make two hands, then we attach the two hands with the body, we make the legs, we attach the legs with the body. That's how the simply clay models are made up. So this is protein threading. We are not making the entire thing at one go, we are making it in an individual section, and then we are stitching them together. That is protein threading. Okay, because it is really easier to model small fragments rather than modeling the entire fragment at one go. It is much easier to model small fragments. Okay, it is simply like biryani. How the biryani is made, you cook the rice, you cook the mutton or chicken separately, you cook the aloo, and then you mix all together and you set it up on a handi and you heat it up. The entire biryani is made. Clear?
Yes, sir.
But in case of fried rice, that is different. You mix everything together and you fry it up. So that is the major difference. Okay. So after the backbone is predicted, then we model the loops, because the loops do not have a proper structure. The loop structure is entirely different. It is absolutely different, right? Because they don't have any proper structure. They can have multiple structures. So later, first we develop the backbone, which is the major structure. Then we model the loops. Finally, we do side chain packaging. Side chain packaging means every amino acid have side chain. Now, some of the side chains are protruding outside. Some of the side chains are protruding inside. Some of the side chains are interacting with the solvent. Some of the side chains are not interacting with the solvents. So that is the packaging, in which way the side chains are facing, which way the side chain are protruding. That is the thing. That's what we do. And finally, the structure refinement is done. Structure refinement means what? You simply bring down the energy. You heat up the system and then you slowly bring down the energy. And as you bring down the energy, if there are certain static hindrance, if there are static clashes, if there are certain bond breakage, then what happens? Those things are elevated. Those things are gone. You get a proper structure which have everything in a proper shape. Clear? So this is the general way of protein prediction. So as I have said you that what is ab initio modeling. In ab initio modeling, you simply start with a particular sequence, only the sequence. So all information you have is about the sequence, nothing else. You only have the sequence. You take the sequence, you feed the sequence to a software or to a server, and the server analyzes each of the sequence, the chemical property of the amino acid. Now the chemical property of amino acid also changes depending upon the environment. If arginine is present inside of lysine, then it is going to behave in a particular way. Again, if arginine is present in front of glutamine, then the nature of that particular thing is changed. So that is how they behave. Not only depending upon the structure, but also depending upon their chemical environment. Okay. It also depends upon the pH in which you are placing in, because the pKa value of all the amino acids are different. Arginine has a pKa value of almost 8. So that means at normal pH 7, it is in a positively charged fashion. It is always in a positively charged fashion. But you simply keep that protein in stomach. The stomach has acid, right? So in acidic condition, the pKa value is again lower. The pKa value changes. So there you don't have a positive charge. Okay. So that is how the protein structure change. Not only depending upon the structure, but also depending upon the environment. Okay. So that is what the ab initio modeling does. Now this particular thing was developed by Anfinsen. Okay. And this is the structure, or this is the method by which the protein in our body folds. The protein in our body folds by ab initio modeling. They don't take a look into how the other proteins are behaving. They simply fold in their own way. But the modeling method, the mathematical model, the mathematical equation that we have to describe this interaction, the van der Waals interaction, electrostatic interaction, the mathematical model that we have to describe those, to define those, is not optimal. Okay, so that's why we don't have a system to predict the model via ab initio model in a 100% correct fashion. Okay.
Yes, sir.
That is why, though this is the best method, but currently due to the lack of those things, we are not able to predict it or not able to use this in a 100% manner. Okay. So after this ab initio method, next what we have is this particular homology model, that in which you have to take a look into the other proteins. So one thing about homology model is that you require the help of other proteins. But in ab initio model, even without the help of other proteins, you can model the structure. And that is more important. Modeling a proper structure without the help of other proteins. But,
So I have
yes,
one question.
yes, sure.
Uh, in the first step is the backbone prediction. Matlab, we can use any of the modeling structure, like
Yes.
it can be homology, it
Yes.
can be protein or amnesia.
Yes. Yes.
I
We
thought
can use.
we have to use the 3 steps itself.
No.
Like,
We can use either of them. Either
okay,
of
sir.
them. Only one of them. Okay. There are 3
Okay, sir.
methods available. These 3 methods, but we can use any of the methods that we feel. Okay.
Okay, sir.
Now
And
currently, yeah, sure. Say.
one more question I have, that is in the stage 4, you said structure refinement. Sir, in the structure refinement, you said that it will check if there is any static hindrance present,
Yes.
like after the entire sidechain packaging and everything is done,
Yes.
it just checks between the bond. That means like,
No. What happens is that in the refinement portion, the energy is minimized. The energy of the system is first calculated. Suppose the energy of the system is 40 minus, or suppose it's minus 44 kilojoule per mole. Okay, that is the energy of the system. So what the software does, it reduces the energy. It reduces the energy further. It simply takes the protein from minus 44 kilojoule per mole to minus 100 kilojoule per mole. Okay, so once
okay,
the software is taking the minimizing the energy, a lot of steric hindrance go away, because there are, this is a small simulation where every protein or every amino acid is moving with respect to one another, and this movement, this particular movement reduces. For example, suppose you are pouring something in a jar. Okay, you are suppose pouring sugar in a jar. What do? After pouring something, we shake up the jar, right?
yes, sir.
When we shake up the jar, what happens? Space is made, right?
Yes, sir.
Why the space is made? Because at first the sugar cubes were resting on one another, but when we shake it entirely, then they find out the other pockets that are there, and all the sugar, they move into those pockets, and that is why we get a small vacuum layer above it, and we can pour more sugar to it. The same thing happens in protein also. You shake the entire system. When you are shaking the entire system, then if there are any steric hindrance, if there are any bonds that should not be there, now see, proper bonds, then what happens? First let me tell this. Then if you shake the entire system, then what happens? Then those bonds, those steric hindrance, they go away. Now you might ask that sir, there might be some illogical bonds, there might be some illegal bonds, there are some bonds that should not be there. How do we break them? They also get broken by that small shaking. For example, suppose you are in relationship with a friend. Suppose you have a friend. Okay, you two are best friend. Now if you two are best friend, and then if I simply come up and say that that other friend was talking this rubbish about you, if your friendship is a true friendship, are you, this small saying, is it going to break your friendship?
No, it's
It is
not.
not going to break the friendship, because you have a very strong relationship. But suppose you casually know someone, you simply casually know someone, your relationship is not that strong. Now someone comes up and says that, yes, I was seeing him or her talking this thing about you. Now if that relationship is not that great, that small talk can break up the relationship, right?
Yes, sir.
It depends upon the strength of the relationship. Same thing, if there is a bond that should not be there, then that means that is not an optimal bond. Then this small shaking can break that bond. But if there was a bond, as I was taking, suppose if arginine 1 should connect to lysine 3, and that is an optimal condition, that happens. So this particular bond is of very high strength. A small shaking is not going to break that. But suppose arginine 1 made a bond with isoleucine 4. Now this is a bond that should not be there. So if I shake it up, then what is going to happen? That bond is going to break, because anything that is not strong enough is not going to last forever. Correct?
Yes, sir.
Okay. So this is why the small structure refinement is done, to see whether the structure that we have received is proper or not. Because if there are even some illogical bonds and all, then they are going to get broken by that structure refinement. Clear?
Yes, sir.
Very good. So in the next class, what I'm going to do, we're going to check and use this particular, we're going to model a particular protein. We're going to model a protein whose structure is not yet modeled. We're going to model the protein, and then we are going to check the structure using the pi model. Okay?
Okay, sir. So we will be using all four of the stages in order to check up protein structure.
Actually, you don't have to do anything. These are the 4 stages. These are done automatically.
Okay.
These are 4 stages. These are done automatically. You simply have to submit the protein sequence. That's done. That's all you have to do.
Okay, so I will be receiving the output from the system.
You are going to receive the output. Okay.
So when will be our next class? Can you please
your next class would be in next weekend. Okay.
Okay, sir.
Yeah. And if I can, I'm going to take one class in the weekday. That I'm going to let you know. Okay.
Okay, sir.
But generally, remember your next class would be, next 2 classes would be in the next weekends. Okay.
Okay, sir.
Okay. And even if it's not in the weekends, I'm going to let you know the. If I let you know the day before, is it okay? When you are asking in the Telegram group that you have to keep it free, is it okay for you to if I let you know the day before?
Yeah, sir. Actually, in the last class, you told me that you will provide the entire schedule in which date you're going to take
Yeah, actually, the thing is that currently there are some issues over at my place. From my end, it's not your problem. And that is actually preventing me to give the schedule. I don't know the correct schedule of myself also because I have to get out of the city. Okay. I'm going to go on there's a conference that I'm attending. So that's why I'm saying that if I let you know the day before, will it be okay for you?
Okay, sir, no problem. Okay, like at least, yeah, if it's in the morning or night, in both the times I'm free. Like if it's, as you said, it's either from 7:30 or 9:30. That time in both.
Yes. If it is in the morning, it's going to be from 7 o'clock or 7:30, and if it's at night, then it's going to be from 9:30 p.m. Okay.
Okay, sir. No issues.
That's for sure, 100%. But the day I'm going to let you know later. Okay. Acha, are you free tomorrow?
Tomorrow, so like if it's in the night, I'm free.
No worry.
In the morning.
Yeah. Okay. So don't worry. It's going to be on the next day. I'm going to let you know. Okay. But most probably it will be on the weekend. Most probably it will be on the weekend. Even if it is not, I'm going to let you know the day before. Okay. I'm going to give you 24 hours time. Head up.
Okay, sir. No problem. Actually, like only tomorrow I'm not exactly free, but after that I'm free. I have no issue with other days.
No issue. Tomorrow I'm not going to take a class. You keep it free. I'm going to take the class next week. Okay.
Okay, sir.
Sure. Thank you. Any more questions?
No, sir. Thank you.
Okay. Sure. Thank you. Bye.
Bye, sir.